So without further delay, I'd like to introduce our first speaker. This evening's first presentation is by Professor Roehe from Scotland's Rural College. Roehe is Professor of Animal Genetics and Microbiome. His overall research aim is to get insight into the microbiome in the rumen, its effects on performance, and its interactions with the host genome. His research involves the identification of quantitative genetic and genome links between the host animal and rumen microbial community, as well as microbial genes to be exploited for mitigation of methane emissions, improvement of feed conversion efficiency, meat quality, animal health, behaviour and welfare traits in ruminants. His collaborative teams have applied bioinformatics to reveal metagenome assembled genomes of rumen microbial species never sequenced before. His publication on bovine host genetic variation and its influence on rumen microbial methane production was awarded the 2017 PLOS Genetics Research Prize based on science, excellence and community impact. In addition, his team's host genetics microbiome research has been shortlisted for the Times Higher Education Honours Award 2018 in the category Research Project of the Year for the STEM subjects, so that's science, technology, engineering and maths. So with that accolade, I will now hand over to Professor Roehe for the first presentation of the evening. So thank you very much, and we all look forward to hearing it, I'm sure.
Thank you very much for the very nice introduction. I want right away to start about the rumen microbiome. The rumen microbiome is a very interesting ecosystem, and I will explain this ecosystem and how this ecosystem is related to methane emissions and improvement of feed efficiency. In my presentation, I will show the link between the microbiome and feed efficiency and how it can be used for breeding purposes. Now, why we want to mitigate methane emissions and improve feed efficiency? Methane is a highly potent greenhouse gas. It is 28 times more has 28 times more global warming potential than CO2. We have livestock emissions worldwide of 7.1 billion tons CO2 equivalent per annum, and 40% of these emissions are from enteric methane, and then the main enteric methane is from cattle. Therefore, there is a big pressure on the farming community to reduce methane emissions from livestock. From the economic point of view, feed efficiency is much more important for farmers because 60 to 70% of the total variable costs are feed costs. And therefore, an improvement of feed efficiency is very important for the profit of farming. But also the reduction of methane emissions is associated with feed efficiency. In addition, the feed is a limited source and should be based on the condition that we need all the feed for our consumption as much as possible, used as efficient as possible. Now is the question, how to improve feed efficiency and methane emissions. To improve feed efficiency, we know animal breeding works very efficient. And to improve these traits, we know animal breeding is very cost-effective. But improvement of feed efficiency and reduction of methane emissions is very costly because it has to be measured. And the cost involved in measuring, for example, methane is so high that we cannot improve it by using methane measurement. We have to use a proxy trait, and we are using the microbiome as proxy trait in order to improve feed efficiency and to mitigate methane. Therefore, I will explain in the following very detailed the microbiome and how it's related to methane emissions and feed efficiency. The microbiome is in the rumen and converts fibrous food into nutrients the animal can absorb. The microbiome is influenced by the host genetic and, of course, by the diet. And we are looking for the optimal microbiome in order to improve feed efficiency and reduce methane emissions. So we are looking for the diets and the host genetics which optimize the rumen microbiome in order to change these important traits. Now more specific, the rumen microbiome consists of bacteria, protozoa, and fungi. These microbial community converts human inedible food into absorbable nutrients. These nutrients are mainly volatile fatty acids, which is energy, and protein, which comes from the microbial protein. And these protein and volatile fatty acids are absorbed by the animal and is used to produce high quality protein in form of meat. Now is the emphasis on these bacteria, protozoa, and fungi. And I want to show you that these microbial community is very dense in the rumen. For example, tens of billion bacteria are program digester. Millions of protozoa are program of digester available, and thousands of fungi. And they are producing large enough nutrients for the animal in order to survive. But there's one disadvantage from the environmental perspective. During the fermentation of food, the methanogenic archaea are producing methane. But this methane is mainly produced due to the fact that there's a surplus of hydrogen during fermentation. And this surplus of hydrogen during fermentation is used by the methanogenic archaea in order to produce methane. So our aim is mainly to reduce this surplus of hydrogen so that the methanogenic archaea have no substrate in order to produce methane. the animal does not need methanogenic archaea. So if we are reducing the surplus of hydrogen, then no methane is produced. In our studies, we are using our facility at SIUC, and we have very nice facility at SIUC to individually record feed intake, to individually record even methane emissions in so-called respiration chambers. But these kind of recording is very expensive and can be done only in experiments. For breeding, we need proxies to predict feed intake, to predict methane emissions in order to use this information on a large amount of animals, because we need a large amount of animals for efficient breeding in order to predict breeding values as accurate as possible. In our experiments, we are using always different diets and different genotypes in order to have a wide spectrum in genetics and also in diets. And we are measuring feed emissions, mainly methane. For breeding purposes, it's very important that the trait has a large variation. And in our experiments, we found for methane emissions a large variation in methane emissions, even if it's the same breed. Here, for example, Aberdeen Angus, and the same diet, here forage, the methane emissions are from 15.9 to 31.4 gram per dry matter intake. So the methane emissions from the low emitting animal to the largest emitting even doubles. So this shows there's a large variation in methane emissions, even under the same diet, the same breed, and the same feed intake. More important is even that the composition of the microbial community, and we are looking at the methanogenic archaea to bacteria ratio, that this composition is different between animals. So here you see for Aberdeen Angus with forage-based diet, Archaea bacteria ratio changed from 1.5 in one animal to 11 in the other animal. So a huge variation in the composition of the microbial community in animals is available. And this huge variation we can use then for breeding, and therefore we found that breeding is very efficient in order to change the microbial community in the rumen, and then by changing the microbial community in the rumen, we can change the traits like methane emissions and feed efficiency. In our experiment, we are looking also always for breed differences, and we found, for example, that Aberdeen Angus had a higher emission per day than Limousin, but the differences are mainly due to a higher feed intake of Aberdeen Angus. On dry matter intake basis, the methane emissions are equal in both breeds. But what we are finding in addition is that the forage-based diet has a much higher methane emissions than a concentrate diet, and we want to use mainly forage-based diet for ruminants and not concentrate, because concentrate diet can also be used for other species. Now we were looking for whether there is a host genetic effect on the rumen microbial community. Therefore, we looked at sire progeny groups. These sire progeny groups are then estimates of the host genetic effect on the rumen microbial community. The rumen microbial community we measured as the methanogenic Archaea to bacteria ratio, and the sire progeny groups. Each group consists of 8 progenies, and these progenies we have taken the rumen samples in order to determine the Archaea bacteria ratio. And as you can see, there are differences in the estimates of the Archaea bacteria ratio depending on the sire effects. That means the host sire has an influence on the microbial community of the progeny of those sires. This shows that there's a host genetic effect on the rumen microbial community. Now we were interested in whether there's also a host genetic effect on methane emissions. And you see here differences in methane emissions of the different sire progeny groups, and that shows that there's a host genetic effect on methane emissions. And now the striking and interesting part is if you see the ranking of these sire progeny groups based on methane emissions and based on the methanogenic Archaea bacteria ratio, you see the sires are equally ranked. So we can use the Archaea bacteria ratio in order to change methane emissions. That means we can use the Archaea bacteria ratio as proxy trait for predicting methane emissions without measuring methane. And it is much more cost effective to determine the Archaea bacteria ratio than to measure methane. We also did heritability estimates, that is the genetic determination of the traits. And we found 40% of the genetic variation in methane emissions is due to genetics. We found moderate heritabilities for growth rate and feed efficiency, and the highest heritability for feed intake. For feed intake, probably because we are accurately measuring feed intake in an experiment, and therefore it has the highest heritability. Now the question is why the host genetics has an influence on the rumen microbial community. So we are interested in what mechanisms are behind it that the host genetics has an influence on the microbial community in the rumen. The rumen microbial community is very dependent on the pH in the rumen. And the pH is depending on the saliva production, because saliva produces bicarbonate, and bicarbonate buffers the pH value. So the pH is depending on the production of saliva. And between animals, there's a large difference in saliva production. On average, an adult animal produces 150 liter saliva per day, and the variation is 25 liters between animals. So therefore, the host genetic effect can be due to the change in saliva production between animals. The differences are also due to short-chain fatty acids absorption between animals, the passage rates of protons between animals, which also influence the pH and is influenced by the rumen, by the host. Oh. The host genetics has, of course, also an influence on the physical size and structure of the rumen. The host genetics has an influence on the contraction and passage rate of the digester, and the faster the passage rate, the different is the rumen microbial community. And there is even a microbiome gut-brain axis. That means there's a crosstalk between the rumen microbiome and the host epithelial cells, which influence the brain. So over this microbiome gut-brain axis, the feed intake, the immune system can be influenced by the gut microbiota. To show this, they have a lot of studies in humans about the gut microbiome. The gut microbiome produce metabolites which influence the immune system, which influence over the endocrine pathways, which influence even over neural pathways, the brain. But it goes also the other way around, from the brain over the HPH axis, over the immune system. The microbial community in the intestine is influenced. That means there's a close relationship between the microbiome and the host, even over the brain. In humans, they know that obesity has an influence from the gut microbiome, and probably over neural pathways, the desire to eat is increased from those metabolites produced by the intestinal microbiome. This shows how close the microbiome is related to the. Now we were interested in getting even better criteria for selection to be used for breeding to reduce methane emissions and improve feed efficiency. And in genetics, the sequence technology to get the whole DNA of rumen microbes sequence has developed in the last years enormously. So we can really sequence the genes of the microbes. And then we get the abundance of the microbial genes in the rumen sample we have taken. And these microbial genes, we know the function of those microbial genes, whether they are in the metabolism of methane production, or whether they are in the metabolism of carbohydrates or any other metabolism. We know which kind of microbial genes are involved in. Now we analyzed in our samples the microbial gene abundance. We identified about 4,000 microbial genes, and here's a network of all these 4,000 microbial genes. Each dot is a microbial gene. And you see there clusters of dots. For example, clusters 4 and 6. These microbial gene abundances are highly correlated to each other. So if one microbial gene increases, the other microbial gene in this cluster also. And then we looked at these cluster of microbial genes. And we found in cluster 4 and 6 mainly microbial genes which are associated with methane emissions. And this shows that microbial genes are increasing simultaneously when higher methane is produced. Then we were looking for those microbial genes which mainly influencing methane. And in the figure below, you see cluster 4. There are 20 microbial genes which are mainly influencing methane emissions and explaining 81% of the variation in methane emissions. And these 20 microbial genes we are interested in breeding in order to predict methane emissions and to reduce methane emissions by breeding. This is a so-called heat map. Here we ordered the animals which we have measured with increase of methane emissions. And on the right-hand side, you see all the 20 microbial genes. And blue means low abundance of these microbial genes. Yellow means high abundance of these microbial. And you see, with increase in methane emissions, the microbial genes are increasing in its abundance. So we can use the microbial genes very efficient to predict methane emissions of animals without measuring methane emissions. We need only the abundance of these microbial genes. And now we found also that these microbial genes are highly heritable. That means influenced by the host genetics. And the heritability of these microbial genes are between 36 to 52%. So these microbial genes are highly heritable. And we know also where these microbial genes are in the metabolism of methane. All these red dots, that are the microbial genes we identified significantly influencing the metabolism of methane. So there's a lot of information in the microbial gene. Now a question is very often, has the diet a more important effect than the genetics, or can we combine dietary interventions with genetic information? And this figure shows we can combine it. We know concentrate has lower methane emissions than forage space, but you see with increase in the abundance of the mcrA a gene, which we know is associated with methane production, methane emissions are increasing in the case of concentrate feeding and also in the case of forest feed. And the increase or the slope is almost similar. That means we can use genetics in order to reduce the relative abundance of the gene, and we can use dietary in addition to reduce methane. So a combination of both dietary intervention and genetics is possible. Also, we were looking at whether microbial genes are influencing feed intake, growth rate, and feed efficiency. And we found other microbial genes, which are different from those microbial genes influencing methane, are influencing feed intake and growth rate. And we can explain high amount of variation of those traits. And this shows again, we can use the microbial gene abundance also to predict feed intake. So the microbial genes can be used as selection criteria in order to predict feed intake without measuring. Now the question, how can we use this information? We have protocols available to take rumen samples from live animals and in the abattoir. Then my screen is a little bit frozen. Then we determine, based on these rumen fluid samples, the microbial gene abundances. And if we have the microbial gene abundances, we can predict the methane emission from the 20 genes we have identified to have an influence on methane emissions. We can predict feed efficiency based on the 20 genes which are associated with feed efficiency. And these are all microbial genes, not host genes. The host genes we estimate based on the host genomics and estimate then estimated breeding values for these predicted feed efficiency from these host genetics. And from the host genetics also based on the predicted methane emissions, the breeding values for methane emissions. And we can do it also for animal health. Based on these research results, I want to conclude that the microbial gene abundance is a very informative trait to predict methane emissions, to predict the daily feed intake, to predict average daily gain, feed conversion ratio. The advantage of this microbiome proxy is that it's much more cost-effective to obtain than to measure methane emissions, than to measure feed intake. And there are thousands of microbial genes, so we can really choose those microbial genes which are highly host genetically influenced and highly genetically correlated with methane emissions and feed efficiency in order to improve genetically these traits. At present, we are working together in a BBSC link project with Genius in order to find a cost-effective methodology to determine the abundances of the Rumen microbial genes. We will develop a microarray where all those genes which are highly associated to methane emissions and feed efficiency are on, so that we get the abundances of these microbial genes, and then we can use the abundances of microbial genes in order to breed animals with low methane emissions and improved feed efficiency. The abundance of these microbial genes can not only use to predict methane emission and feed efficiency, it can also be used to predict the healthiness of animals. For example, acidosis, susceptibility of animals could be predicted. We can predict the resilience and robustness of animals. We can predict meat quality of beef, mainly the fatty acid profiles of beef can be predicted by the microbiome. Probably also the reproduction efficiency of animals. Our research is funded by the Scottish Government, by BBSSC, by the breeding company Genius, aligned research. We have funding from Quality Meat Scotland, from DEFRA and AHDB. I'm working together with colleagues from the Roslin Institute, the Roehe Institute, and our research was very successful. And now I'm happy to answer a question. Thank you very much for your time.
Lovely. Thank you for that. That was really informative. And we've had a few questions come in. I don't have time to read them all out. However, I've selected a couple. So the first one I'm going to read out is from Kate Williams, and it says, what sort of role do you anticipate the Rumen eukaryotes playing in relative abundances with regards to methane? We know the protozoa, holotrichs in particular, produce hydrogen, so it would seem likely that this would result in more methane. So might low methane emitters produce fewer holotrichs?
We did a nice study about the Rumen microbial community and which kind of community plays a bigger role on methane emissions. And what our research now is showing is the abundance of the bacteria, the abundance of fungi, and also the abundance of protists and protozoa has an influence. So it is more important to change the bacteria community, that there's no substrate produced by this bacteria fermentation of hydrogen for production of methane by the methanogens. So our research more and more shows that bacteria, fungi, and protozoa are more important than the methanogen itself in order to reduce methane by breeding.
Lovely, thank you for that. I have another question here which relates to wild herbivores. Is there any data on methane emission conversion rates from wild herbivores, and if so, how do they compare with domesticated ones?
Wild, there have been information about methane emissions in deers and in all wild animals. Is that the question? Yeah. in wild animals. So there are differences in the community between these wild animals. One interesting part is probably the kangaroo. The kangaroo has also rumen, but it does not produce methane at all. And therefore, people are always interested in what's the kangaroo doing differently than other ruminants. And this shows on the other side, we do not need methanogens in the rumen to produce methane. The cow can live without methanogens. We have only to make the rumen microbial community more efficient so that no surplus of hydrogen.
Thank you for that. I'll do one more question because I know we are running over time and I don't want to upset my co-chair, Will. So, okay. I'll just ask one more quick question. And I suppose this relates to the ground level impact of your research. And I'm going to combine two questions actually into one, which is that, is the output of your research already having an impact at the grassroots level on farm? And if not, how long do you foresee that taking? And the second part of that, I suppose, is, this is a ground level question, which is better, higher, low pH, presumably low pH is better, so is less saliva better? And in which case, could we produce supplements for on-farm use that could potentially reduce pH in that direction?
There are possibilities to use supplements in order to change the rumen microbiome, and there's always an optimum kind of pH in order to improve feed efficiency, in order to improve animal health, and also to reduce methane emissions. We know, of course, that the lower the pH is, the more methane emissions taking place. The first question was, just shortly?
Yes, so how long do you foresee before this has an impact at the ground?
Yeah, we are working together with the breeding company, and we hope, of course, to have a big impact due to this association. They are paying a lot of money, so they would not do that if they're not expecting a lot based on our research. And the present kind of results we are getting are quite promising. And of course, to implement it, it needs to build up this microarray, and that needs time. So it's still some years until we know this is heavily implemented. But I hope the implementation time is shorter because we are working already with the breeding company.
Lovely, thank you so much for that. Obviously, in normal circumstances, you would be in a room and not virtually, and there would be a round of applause. So the silence does not mean that obviously this wasn't a great and informative presentation. So on behalf of everyone listening, thank you so much for taking part, and thank you for your presentation. And I'm now going to hand over to Will to introduce the second speaker. So thank you again.
Thank you.